A very good morning to all of you. So in today's session, we will continue our yesterday topic, that is spectrophotometer. Yesterday, we have studied what is the use of spectrophotometer, what is the application, what is principle, and how many types of spectrophotometer. Okay. If you don't watch that video, so you first watch that video so that you can easily understand today's session. Today we will study what is the instrumentation of a spectrophotometer. Okay. So in a spectrophotometer, what things is there? First thing is your light source. Okay. First is your light source. After that, there is a second thing is there is a lens or also known as collimator. Third is your monochromator or monochromator is also known as prism. Then fourth one is there is diaphragm or wavelength selector. Then there is a cuvette. After that, photocell is there and there is a display. Okay. So in the instrumentation of a spectrophotometer, there are 7 things: light source, lens or collimator, monochromator or prism, and diaphragm or wavelength selector, then your cuvette, photocell and display. Now we will discuss how these will act and what is the function of all these. So light source. What is the function of this light source? This light source provides what? This light source provides the light. Okay. What is the function of this light source? The light source is there, and for measure the concentration, we should need light, na, particular wavelength of light. So the main function of light source, it provides the light. Okay. Then what is the function of this lens or collimator? The function of this lens and collimator, it transmit the straight light. Okay. Light generated from this light source, okay, and then it will enter into your lens or collimator. And what is the role of this lens or collimator? It will transmit the straight light. Okay, transmit the what? It will transmit your straight light. Okay, this is the function of your lens or collimator. After that, light passes through where? After that, light passes through this your prism or monochromator. What is the function of this prism or monochromator? It will split the light. Okay, when light will enter into monochromator, then it will, what is the function? It will split the light. That particular wavelength of light, it will split the light. Okay. After that monochromator, there is a slit or diaphragm, or it is also known as wavelength selector. As the name suggested, wavelength selector, that means the function of this wavelength selector is it will select a particular wavelength of light and it allowed to pass a particular wavelength of light. Okay. Have you seen it? The function of this monochromator, it will split the light. But when it passes through this, what? When it passes through this diaphragm or wavelength selector, then this will select or passes on a particular wavelength of light. Okay. So, what is the function of this wavelength selector? Select particular wavelength. Select particular wavelength of light. Okay, select particular wavelength of light. So, this is the function of your diaphragm or wavelength selector. After that, light passes from where? After that, light passes through your cuvette. Cuvette is what? Cuvette is nothing but it is a what? Cuvette is nothing but it is a kind of small test tube. And we have studied yesterday topic that the material of cuvette depend on which type of like spectrophotometer you are using. Whether you are using visible, whether you are using UV rays, or whether you are using infrared. According to that, the material of cuvette depends on that. Okay, yesterday already we have discussed that. Okay, so in this cuvette, what is there? In this cuvette, there is a sample. This is your sample. Okay, this is your unknown sample whose concentration you want to measure. And in this cuvette, you will fill that your sample. Okay, for which you are willing to measure the concentration. Okay, so this cuvette contains your sample, and then this particular wavelength of light passes through this sample, and when the light passes through sample, then what will happen? What will happen over there? Beers-Lambert law. Beers and Lambert law. Applicable over here. Okay, when light passes through that, then Beers and Lambert law is applicable over there. That is your Absorbance is directly proportional to the path length of light and concentration of light. Okay. So that it will absorbance will is there. When light passes through there, then according to this Lambert speed law, this absorbance will depend on what? This absorbance will depend on your path length. Path length is this length. Okay, this length the light travel when it passes through your sample. This is the path length. This one is your L. Okay, L. And absorbance is directly proportional to your path length and absorbance is directly proportional to your concentration. Okay, according to that particular wavelength when it passes through the cuvette sample. Okay, absorbance will be there. Then what is the role of this photocell? This photocell converts your heat energy. What is the role of this photocell? It will convert your heat energy into your electrical energy. It will convert heat energy into in the form of your electrical energy. Because in the detector or display that will show the reading, that cannot show the heat energy. So it is important to convert the heat energy because from here the energy which is generated is heat energy. So the role of this photocell is to convert heat energy into your electrical energy. Alright. And when it passes through the display is also known as galvanometer. Then what is the function of display? It will show OD, that is optical density of your solution. Okay, OD, that is what is the full form of OD? Optical density. Okay, then finally this display will display your instrumentation is there and there is a display in which that reading will come. Okay, finally after this process that display will show the optical density of that unknown solution, and from that optical density we can easily calculate the concentration of this unknown sample. Alright, so this is the whole instrumentation of your spectrophotometer. Clear? So if question asks spectrophotometer, what points you have to mention? First you will write what is the use of spectrophotometer. So what is the use? Spectrophotometer is used for what? It is used to find out the measure the concentration of any unknown sample. Okay, this is the use. After that you will write the principle. It is based on the Lambert speed law. After that you will write how many types of spectrophotometer, that is based on the beam of light, and based on beam of light there is a single beam and double beam, and based on the what is the source like visible, UV, infrared. Already we have discussed that. After that you will write the application and then you will make this diagram in your exam and you will write all this instrumentation. You will write it in that way only you will get the full marks. Okay, you will just make this diagram and in lower side you will write the functions. Okay, so this is the complete topic of your spectrophotometer. Till here it's a complete for UG student, but for PG one additional question in this topic is asked sometime. Okay, that is what is difference between single beam and double beam spectrophotometer. So for PG student you must know the difference between single beam and double beam. For UG up to this it's only you have to write in your exam. Okay, but for PG student you have to know the difference. Okay, so what is the difference between your single beam spectrophotometer and your double beam? Single beam and double beam. So first you have to know what is single beam. The diagram which I have shown here previously is your single beam, because here only one beam, that is single, that is only one beam is passes through your cuvette solution. But in double beam there are two beams. Okay, there are two kind of slit or diaphragm is there, and by these two slit or diaphragm two beams passes, and one beam is passed through your sample solution, that is your unknown solution. Okay, and other beam of light is passes through your blank solution. Okay, so this is difference between single and double beam. Then what you will write in your exam? First, it is less expensive. Okay, single beam is less expensive. This is for PGs only. Okay, PG students only. Okay, it is less expensive and it is more expensive because it is more complicated, more instrumentation is there. So double beam is more expensive. Okay, Second, it is simpler in structure. Simpler in its structure, okay, while it is complicated, or you can write it is complex, okay, because there are two beams are there, one blank solution is there, two cuvette you will use. So because of more instrumentation, it is more expensive, it is less expensive, and it having only simple structure, but it is somehow complex structure. Third, it is less accurate. Accuracy is, if you talking about accuracy, then single beam is less accurate and double beam is more accurate. Okay. So question can be asked like why single beam is less accurate and why double beam is more accurate? Why? Because the reason is that sample solution and your blank solution. Sample solution and blank solution we use separately. So there is a chance of error. So there will be more chances of error. That's why it is less accurate. Why? What happened in? It is more accurate because here what happened? Your sample solution and your blank use simultaneously. So less chance of error. Once again, we will understand that while it is less accurate, while it is more accurate, because in single beam there is only one beam passes through. So first one beam is passed through your sample solution, then you will calculate the OD of your sample solution, then again you will just put all kind of instrumentation, and in a cuvette you will put blank. So you will do two separate processes. First you will measure the OD of your blank solution, then you will measure the OD of your unknown solution. Okay, so if you will do some process separately in two times, then there is a more chances of error. But why? In double beam, what is there? Simultaneously, like there are two beams, and you will put together your blank solution and your unknown sample. Okay, and two beams are passing. So one beam is passed through your sample solution of unknown sample, and other beam will pass through your blank solution. So simultaneously you can measure the optical density of your that sample solution and your blank. So there is a less chance of error, and because of less chance of error, it is more accurate. But it is more expensive because it is a complex and it need two beams, it need two cuvette together, while it is less expensive and it is simpler in structure. So this is all for your university exam for UG and PG. Spectrophotometer, it is asked as a 10 question, can be asked as a long question. If long question will come, the question can become as a what is the use of a spectrophotometer, what is the principle of a spectrophotometer, writes its application along with instrumentation. And for PG, sometimes question can be asked as a what is difference between single beam and double beam. Okay, and in the next video we will discuss one more thing. Sometimes question can be asked in Viva and theory also, what is difference between spectrophotometer and colorimeter. Okay, so that you also have to know, that is what is the difference between colorimeter and spectrophotometer, and that question is important for both UG as well as PG. So in next video we will discuss spectrophotometer and colorimeter difference. Okay, and for today this is all for your this question. Thank you.